Important MCQs in biotechnology and process. 2 microbes found to be very useful in genetic engineering are Escherichia coli and Agrobacterium tumefaciens, Vibrio cholerae and bacteriophage, Salmonella and cocci, Bacilli and gonococci. First choice is correct. Escherichia coli and Agrobacterium tumefaciens are the 2 microbes used in genetic engineering. Restriction endonuclease. The function of the enzyme restriction endonuclease is cuts the DNA molecule randomly, cuts the DNA molecule at specific sites, restricts the synthesis of DNA inside the nucleus and synthesizes DNA. Restriction endonuclease. This is an enzyme produced by the bacteria which is used in genetic engineering to cut the DNA molecule at specific sites. DNA has got 2 strands, so to cut at the desired site, the restriction endonuclease enzyme is used. So second choice is the correct answer, cuts the DNA molecule at specific sites. Human insulin is being commercially produced from a transgenic species of A. Mycobacterium, B. Rhizobium, C. Saccharomyces, D. Escherichia. Fourth choice is correct, Escherichia. From this species, human insulin is commercially prepared. What we are using now for diabetic patients are commercially synthesized from this species, Escherichia species. DNA or RNA segment tagged with a radioactive molecule is called A. clone, B. plasmid, C. vector and D. probe. This is called as probe. So DNA or RNA segment tagged with a radioactive molecule, you call it as probe. They are used in the screening stage to identify specific sequence of DNA in a mixture. Okay, to identify specific sequence of DNA. The sequence of DNA in a mixture is identified using this probe. Restriction endonucleases or enzymes which restrict the action of the enzyme DNA polymerase, remove nucleotides from the cut ends of the DNA molecule, make cuts at specific positions within DNA molecule, recognizes a specific nucleotide sequence for binding of DNA ligase. C is the correct choice, makes cuts at specific position within a DNA molecule. Given below is a sample of portion of DNA strand. Okay, this is one strand and this is the complementary strand. Giving the base sequence on the opposite strands. What is here special about? See the bases guanine, adenine, thymine, cytosine. Here the exact is repeated here, cytosine, thymine, adenine and guanine. This is called as palindromes. Okay. The base on the complementary strand will be an exact repetition in the reverse order. This is called as palindrome sequence of base pairs. So the answer, let us see the 4 choices. Replication completed, deletion mutation, start codon at the 5N and palindromic sequence of base pairs. 4th choice is correct, palindromic sequence of base pairs. There is a restriction endonuclease called EcoR1. What does co part in it stand for? This is stand for the coli. The generic name EcoR1. Okay, so E is the generic name, and this is the coli is the species name. And R is the particular strain which is used, and 1 is the first enzyme which is extracted from this strain. And next question, agarose extracted from seaweeds used in gel electrophoresis. This is a direct question used in DNA extraction, separation and isolation of DNA. This is given in NCERT. This is a direct question. Which one of the following technique made it possible to genetically engineer living organisms? A. Hybridization, B. Recombinant DNA technique, C. X-ray diffraction, D. Isotope labeling. Second choice, recombinant DNA technique is the correct answer. So with the help of recombinant DNA techniques, we can genetically modify the living organisms. Which one is a true statement regarding DNA polymerase used in PCR? It is isolated from a virus. It remains active at high temperature. It serves as a selectable marker. It is used to ligate introduced DNA in recipient cells. Which of the following is true? They have asked which one is true. So it remains active at high temperature. All the other 3 statements are false statements in relation to DNA polymerase.